that it's the most important thing in my career has been to train, mentor, and help facilitate other people's growth. One of the things we're suffering today in science is that we're in a world where the anti-science has become so common. Trying to develop the first immunotherapy for type 1 diabetes taught me a lot.
Hello, everyone. Welcome back to OncoDaily. And today it's a great honor and pleasure for me to welcome Professor Jeffrey Bluestone. I don't think he needs introduction in the professional community and beyond, but we are going to make talk about his career and about the story of his success. So I'm not going to be going deeper. Thank you very much for having a time for us. And just from the beginning, how it started from early days.
Early days. Well, it goes back quite a while. I was an undergrad at Rutgers University and had a chance to do a senior project with a professor there, Robert Cousins, and it was a transformative experience for me. I found the opportunity to really dig down and do research and understand a very deep problem. At the time, this was in nutrition and how molecules bound to metals. It actually turned out it was calmodulin when they finally discovered what it was, and he was very successful. But for me, it really started as really this inquisitiveness of trying to understand things we just didn't understand. But I wanted to apply it to human health. So I ended up going to graduate school, first at Rutgers in virology, but then ultimately at Sloan Kettering Institute, where I really focused on cancer immunology. This was back in the day when people didn't have a lot of confidence that the immune system would be able to tackle cancer. A guy named Bob Good was running Sloan Kettering at the time, and I went to work with him, and he was really one of the first cancer immunologists. Actually had gotten his start discovering the cause of bubble babies, the immune deficiency in people. So I worked there. Then I went on to the National Cancer Institute, again planning on continuing my work in cancer, but got sidetracked into organ transplantation. Why? Cancer was a real black box at the time. We knew very little about what it meant to be an antigen, something that a T cell would recognize in a tumor. We really didn't understand when that exposure took place. Whereas in organ transplantation, it was very straightforward. You knew when the tissue was going in. You had a good idea of what was being recognized, the major histocompatibility complex. And I felt that it was an opportunity for me to really dissect and understand better the mechanism of antigen recognition. I moved on from there to autoimmune disease at the University of Chicago, where I also was working on cancer. Was working on areas like checkpoint inhibitors, as an example, CD28, and then ultimately went to UCSF, where for 25 years I ran a lab. But I also had an opportunity to head up a number of consortiums, including the Immune Tolerance Network, the Parker Institute for Cancer Immunotherapy. And then ultimately, about 5 years ago, I moved and started a company to try to develop some of the work that I had started at UCSF towards a product. So it's been a long, winding road from cancer to organ transplant to cancer to autoimmunity. And the common theme throughout all of it was immune tolerance. How do you induce it and how do you break it? And understanding the rules and how to develop drugs based on that was really key to a lot of the work that I did throughout my career.
What was key to your success?
Oh, I don't know. That's a subjective. I guess I've been successful. I think the key. I always say there are three things mantra that I have written on the wall here. Kick-ass science. I mean, I think first and foremost, you try to do really impactful science and make sure that it is important. Second, collaborate like hell. I've always been Very, very convinced that collaborative science is always more successful than individual science, and so been very fortunate through the consortiums that I've led, as well as my own efforts to work with others, sharing reagents. We made the first anti-mouse CD3 antibody, which led to the development of a human anti-CD3 for type 1 diabetes. But those early days, I'll never forget a meeting I was at a Gordon conference that I was at, and I was young. This was 1985, and I got to go to this meeting, but the meeting kept running long and longer and longer, and it was now 10 o'clock at night, and I'm the last speaker in the last session, and finally I get called up to talk. So I went back to the slide projector. In those days, we had slide projectors, and I told the projectionist, just put one slide in the carousel, and I showed the slide, and it was my name and address. And I said, we've made an anti-CD3 antibody. I'm sure a lot of you will want it. So here's my name and address. Just write me. I think I sent it out to a couple hundred people after that. So collaboration's been key to my success. And then the last is sort of a commitment to make a difference. There are a lot of things we can work on. They're interesting. They're important for sure. But to me, I've always wanted to do things that I thought could make a difference, either in a student's life or in a patient's lives or in a colleague's and life. And so I think that commitment has worn well.
Who were your mentors and what did you learn from them?
So, as I said, I would say Bob Cousins was amazing. He really got me into science. But probably my most influential mentor was a guy named Frank Fitch. And Frank was the head of the immunology group at University of Chicago when I moved there. I had been at the NIH for 8 years, had done well, but decided that I had bigger horizons, and so moved to University of Chicago. And I'll never forget this. Frank sat me in his office and he said, we can sit here and we can negotiate a deal, and I'll negotiate it with you down to the penny of what we'll get. But in the end, you need to do what you want to do. And so whatever the bottom line number is, ignore what you budgeted. Just follow your nose, do what you think is going to be important. That combined with a level of honesty and transparency, and I've tried to build my career on being as honest and transparent as possible, because at the end of the day, trying to keep things a secret, think you can somehow win if you don't disclose. I found that, and Frank was the ultimate honest gentleman from the Midwest, and just a good guy. So he was probably my most influential influencer.
And who are your mentees? I mean, I'm sure throughout your career, either as a scientist or as an innovator and entrepreneur, or also like you had a lot of administrative roles at the university as a provost. So, I mean, what are your mentees?
Yeah, I've been very lucky. I've had over 100 students and postdocs. Overwhelmingly, they have done extraordinarily well. I really pride myself in having been involved with these great scientists and human beings. It's hard to name anyone. I mean, so many have done well. Professors at universities, heads of departments. My first postdoc, Rafi Hirsch, is now head of pediatrics at UCSF. I have a graduate student early on, Randy Crone, who is head of the Department of Pediatric Rheumatology at University of Alabama, Birmingham. But I've got faculty everywhere, and I've got people who've gone to industry and made really important contributions in industry. There's a whole group of people that I trained at University of Chicago, Marissa Allegre, Ann Sperling, others who have done exceptionally well. So I think from the student mentorship perspective, I can't be more proud of the people that I've been able to at least think that I've helped train them. Of course, when you're also the head of a department, which I was both in Chicago and in San Francisco, you get to bring young faculty in. And I also feel like I've been very fortunate in being able to shape and help shape some of those folks. I mean, Mark Anderson at UCSF has done extraordinarily well. He's just a superstar, and he was my first recruit when I got to UCSF. I've had similar recruits at University of Chicago who've done exceptionally well. So it's hard to, I mean, I could name people for 20 minutes, but there's no doubt that it's the most important thing in my career has been to train, mentor, and help facilitate other people's growth.
There is a common belief that, like some kind of opinion that if you want to be a successful scientist, you need to devote yourself entirely to that one, and you're not going to succeed in the other parts. But you were, I mean, successful in different parts, not just only in the science, but also as an administrator, as a like entrepreneur. I mean, how you were balancing this? And
Yeah, you don't want to talk to my wife or my kids. It would not come off well. You know, it's funny. I think the core principles that I laid out earlier, if you keep them in sight, no matter what job you have, you know, and when I was any kind of leadership role I had, this concept of doing great science was so important. So if it wasn't my students or our work, it was someone I recruited or a colleague or as provost being able to support the people who were doing great science. I mean, if that's an underlying principle, the job actually is easier because you basically are always focused on the same thing. Collaboration, again, something it works in my lab when people were working on projects that they could be synergistic with each other. It works in a university, it works in a department. So a lot of the jobs that I've done that are sort of a common theme, and therefore I think easier to do more than one at the time. I would say I've also benefited from try to say this well. I've benefited from being a pretty good salesman. You know, we discount that when you're in science. You know, it almost sounds like a bad thing, but the reality is whether it's a group of eighth graders or it's graduate students or it's the funding agencies, you've got to be able to tell your story. You've got to be able to articulate why you think what you're doing or what's being done in your department or whatever is important and matters. And I think I've had a pretty good ability to do that. My father was an insurance salesman, so maybe he taught me well. But I think that's one of the things we're suffering today in science is that we're in a world where the anti-science has become so common, and as scientists, we don't know, some people are gray, like Eric Topol and stuff, but for the most part, we don't know how to talk to people about science in a clear, succinct, simple way, so they appreciate the value of science. And I think we as scientists can do a much better job than we do of being able to articulate what we do in a way that anybody can understand it, in a way that can be impactful, in a way that really highlights why does discovering restriction enzymes, you know, change the world? Well, it led to the cloning of insulin, and which helps, you know, hundreds of millions of people. So if we could do a better job at telling our story, I think we'll be more successful. And I think I've been pretty good at storytelling.
so, I mean, what's your I mean, what would be your lesson to teach the other scientists or everyone in the sales regard?
Yeah. Yeah, I think part of it is making it simple. If you can't tell the story, and I usually use as a metric, you know, a science major, you know, in high school, someone who wants to go inside. If you can't tell your story to that individual who's not had deep understanding of the immune system or whatever, then you're going to have trouble. One of the courses I taught with Frank Fitch was a great course at University of Chicago. It was immunology for non-science majors. And so I'd walk into class the first day and I'd say, you know, my daughter has chicken pox. Should I be here teaching you today? Or which is, you know, more concerning to you, herpes or HIV? Things like that, questions that provoke people who don't have a deep understanding. So I think that we have to keep it simple. We have to keep it interesting. We have to tap into people's lives that they're living every day. And then, of course, in the case of things like writing grants or philanthropy or even raising money for my company, is helping people understand the opportunity, the value, and the difference that it can make in people's lives.
What was the criteria for being your team member? How you were selecting people?
Well, I probably was over the years a little bit tougher that in retrospect I used to ask people really deep questions. My postdoc that I had done with David Sachs, when I interviewed with him, the first thing he did was he put me on a blackboard and he said, "Okay, I want you to explain the genetics of an inbred mouse or something like that." And I said, "Really?" Yeah, draw it out on the blackboard. So I think that I have been, when I identify people, I put a very high bar in terms of being smart, being effective communicators, which we've already talked about, and being committed and passionate about what they are doing. And again, I overwhelmingly have been lucky, and the choices that I've made have been good ones.
Going to the Parker Institute for Immunotherapy, Cancer Immunotherapy. What was the story? How it started?
Yeah, that's a great story. So Sean Parker, who hopefully your audience knows, having developed Napster and was a really amazing, smart guy.
Facebook CEO, yeah.
Yeah, and Sean came to it. I was provost at the time, and came to my boss, the chancellor, Sue Desmond-Hellmann, who had come from Genentech, had a product development, an amazing leader, an amazing person. And he said, "I am committed to cancer immunotherapy," which he said, "Really?" He said, "Yes. I've read the literature. I know what's going on in the field. I've been to Fred Hutch in Seattle. I've been to Sloan Kettering. I think that we're going to be able to cure cancer using the immune system. Period. Stop." And she said, "Okay." Now, she was an oncologist herself, and so she knew a lot. She said, "Well, what about other cancer drug approaches? I worked on antibodies to HER2, and we cured that cancer, and I worked on other cancer drugs." He said, "Nope, immunology. It's going to be immunology." And so she said, "Well, Sean, if you really want to understand immunology, you've got to talk to my provost." So she walked him over to my office, and we sat down and probably talked for 5 or 6 hours about his vision and about what he was thinking. And I talked to him about how I thought we could execute on it. And then we spent time talking about the model. What kind of model could we create that would basically do the kinds of things that I've been talking about, how we can make sure that the best science in the world is being done, and we would be able to collaborate like hell and make a difference. So the best science meant that you had to go out to the institutions and people that were doing the best science. And that's how the Carl Junes and the Jim Allisons and the Louis Laniers and Tony Rebus and all these folks got involved because they were just doing spectacular science at great institutions. So we brought them on. And then how do we allow them to continue to do great science? Well, that was, you build a sandbox, and this was our sandbox. It had walls. Not everybody could be in the sandbox, but if you were in the sandbox, that was great. But you had to had sand toys and things for people to use in the sandbox. So we built a big bioinformatics group. We built a clinical trials group. We built a infrastructure so that those great scientists would be able to get stuff done. And then the last was how do you make a difference in collaboration? And I just thought that's going to be, we're going to have these retreats, and we're going to have these retreats where it's, instead of people, Nobel Prize winners and the like, flying in, giving their talk and flying out, we were going to set it up in a way that everybody would want to be there. So Sean said, well, let's do it in a really nice place. And so he picked great places to have the retreats. Let's let people bring their families. Let's have people fly business class. Let's make sure that we provide a sandbox that people want to hang out in. And people would come to that meeting, 100 plus people, and they'd stay the whole time. And they'd show their best science, even when it was not published, when it was just early days. Because first of all, if you're sitting next to Carl, you're not going to show them some mediocre stuff. You're going to show the best stuff you got, right? And so the retreats ended up being a magnet for, I think, great ideas and great collaborations and great science. And then the other idea that Sean had, which I thought was fantastic, was, we shouldn't let it stop at the bench. We should figure out a way not just to get into patients, do that, but get it out to the world. And so he, in his entrepreneurial spirit, built an infrastructure that allowed us to both get access to intellectual property from the universities, but then form companies. And I don't know how many companies Pisces has formed at this point, is probably a dozen plus. But that's also, I think, another way to make sure that you make a difference. And he was really good about that. So it was a very interesting model. I think it's worked very well over the last 10 years. And I think that it's really allowed us to make an impact in a field, even though we're relatively small compared to the likes of big pharma or other companies.
Was it easy to work with Sean?
I find it's always easier to work with really smart people, and he's a really smart person. Did I have to take calls sometime at 2 or 3 in the morning because he'd have an idea and he'd call me up? Yeah, I probably would have wanted to have less of those. But overall, I think Sean is just such a committed, dedicated person, and you don't usually find in philanthropy. And I have enormous respect for the people who've given, someone who is as passionate and committed about the science. So Sean and I would spend hours talking about the science, and he'd read the papers, and he would go to figure 3 and stuff. And so that combination of, commitment and passion with the smarts, it's hard not to be compelled working with him. As I said, sometimes his eyes were bigger than my stomach, and his ideas were more than I think we could handle. But for the most part, it was fantastic.
Next year, it's 10 years already, right? Since the establishment.
Yeah.
And what do you think it's going to be the future of the Institute?
Well, the good news is that, during COVID, things, I had left. They didn't replace me with another CEO, and COVID, made it difficult to get together and do things. So it was a 3-year period in which things, but very recently in the last year, they brought on a new CEO, Karen Knudsen, who's doing a great job. She had been the head of the ACS, and she's fantastic. She's out there. She's really telling the story of Pisces, which is great. And they recruited Ira Mellman as president. And of course, there's no better cancer immunologist and scientist than Ira. So I think that bodes very well. It's a renewed commitment to the Institute. I was just on a board call a couple of weeks ago, and a very exciting new strategic plan that Karen and the team put together. And Sean was on the call and very, very engaged. So I have very high confidence that the Institute is going in the right direction, and that it's really going to build on the successes that we had early on.
Yeah, I'm going to interview tomorrow, Karen, by the way.
Oh, great.
What question should I ask her?
Well, if she's willing to share some of her ideas around the strategic plan that they're just rolling out, I mean, it's exciting, and you should hear about what some of those plans are. I also I've never talked to her about this, but I think getting her perspective on the difference of running a very large organization like ACS and this, small enterprise. I'd be interested in her thinking about what are the differences and how it affects her ability to strategize around it. And I guess I'd also ask her, how because she was responsible for cancer writ large at the ACS, and where she thinks cancer immunology fits in, cancer immunotherapy fits in the world of cancer treatments and science.
What would be your advice to the younger generation?
Yeah, it is a very difficult time right now. I will say that I have tremendous empathy for young people trying to get into the field right now. All the barriers that have been put in place, from financial barriers to visas to even the general perception in the public, as I said earlier, about science, I guess I would tell them to hang in there, to continue finding ways to continue their training, whether hopefully it's in the U.S. when it fits, if not somewhere else, to be willing to use their voices in support of science, because that generation is, us old guys, we're going away, and the next generation's really going to have to take on the mantle. And so I hope people still continue to go into science. I hope that trainees see value. And then the last thing I would say is that what I have learned in the last few years, having started a company and running a company, is that science can be done in a variety of different settings. It can be done in universities. It's where great science has always been done. It can be done in small biotech. It can be done in larger pharmaceutical companies. If you have the right people and you have the right commitments by those groups, you can do great science anywhere. And so don't start out with any preconceived notion that there's only one way to do the science that matters, because I think things are now in a place where you can make impact in a variety of different settings.
Was there a moment or a story which resonated most with you throughout your career?
Wow. Yeah, I don't know that I have a story that resonated. I will tell you the journey that I and my colleague Kevin Harrell took in trying to develop the first immunotherapy for type 1 diabetes taught me a lot. It taught me about how important the work was we were doing, because so many of the people affected were kids, and meeting with their parents and hearing the stories. Everyone says, oh, diabetes, you take insulin, you're fine. Well, the answer is no. It's a terrible disease. It's devastating for families, and hearing these families talk about the impact on their lives and how it affected them, it motivated me. I think hearing the stories about how hard it is to develop a drug with all the barriers from industry and the FDA, and actually having taken 35 years from the time I made the drug till the time it was approved, I learned a lot, and I heard the stories about how it doesn't matter necessarily whether the drug works. Does the economics make sense? Does the requirement for infrastructure, for screening, whatever it might be. So I think the stories that have resonated with me over the years have been the stories of the challenges and difficulties, and how you need to be resilient, and you need to be persistent, and you need to be committed, because, you're going to get a lot of things in your life that just are constantly putting barriers in your way. And so to me, the stories were about that kind of persistence and resilience and commitment that people, patients, drug developers, everyone has to have. So I think that's the thing that sticks with me the most.
What about now? What are you focused on? What are you doing at your company?
Yeah, so the company, Sonoma Biotherapeutics, grew out of work that I did in my lab at UCSF with Kishi Tong, a co-founder. And it was built on the seminal work by Fred Ramsdell, Nobel Prize winner, and Sasha Rudensky at Sloan Kettering, understanding the basic biology of a small population of white cells called regulatory T cells. And these cells, we've learned over the years, are fundamentally responsible for peripheral tolerance. And I told you my career has always been about tolerance, how to make it and how to break it. And Tregs are the kingpin. They're the ones that really determine it, because they surveil the body and they look for autoimmune or inflammation and they shut it down. So back starting in the early 2000s, when we first started working on these cells, I believed that we should be able to make a therapeutic for these cells. And, how you do that. Is it a small molecule? Is it a biologic? Well, we decided to go big. So we decided we were going to build a cell therapy. And so we came up with ways of isolating these cells, of growing these cells. Ultimately, in Sonoma Bio, we actually put genes into these cells to make them specific for an antigen. And we give them back to the patients in the hope that they'll home to the right place, that they'll produce the factors that regulate inflammation and autoimmunity locally and impact the patient. We're recently at the American Academy, the American College of Rheumatology, presented our data out of our first trial in rheumatoid arthritis and showed very significant clinical effects of our cells in patients with RA. And so it's been a long road, but I think there is a path towards a cell therapy with Tregs. There are other companies involved in this in various aspects, whether it's autoimmunity, organ transplant. But I do think that we have an opportunity here to really create a new modality of how do we think about living drugs? How do we create long-term tolerance without having to retreat with drugs every week, month or so? And very excited about where Sonoma Bio is right now.
What were the books which you would recommend everyone to read?
Yeah, so when I was at Sloan Kettering, there was a Lewis Thomas was the president of Sloan Kettering, and I got to hear him speak when I was this young scientist growing up. And he wrote a couple of books, The Life of a Scientist, other books in which he really talks about what it's like to grow up in science and be a scientist and be successful. And I would recommend any of his books. He was a tremendous communicator, for sure. And then, of course, like everybody else, the most impactful book for me was Gene by Lewin, Ben Lewin, which I remember sitting on the lawn in Quebec, Canada with my wife, who we had just gotten engaged, and we were traveling in Quebec, and I was sitting on the lawn of the Frontenac Hotel, and she wanted to get some wine, and I was reading Lewin's Cell because I had my prelims coming up when I returned. So yeah, that was an impactful book for me.
Can you tell a bit more about your family?
My family. We don't have I don't have a very interesting story. My family was great. Very, very much a family unit. My brother, sister, and father were all in the insurance business. And my mother unfortunately died very young, of heart disease, when she was in her early 50s. I think the thing that I most remember growing up, my father at the time was a dry cleaner, and we would go on Saturdays. I would go into the dry cleaners with him, and I would help out. I would mop the floors, and I would do a variety of things. Sometimes I tried my science out, and so I would mix bleach in with other combustible things and start a fire. But other than that, for the most part, he kept me there. And I remember being in the dry cleaning store because it was not, he wasn't a lawyer. He wasn't an MD. He was a dry cleaner, right? And so it all was about people. And whether it was the people who worked for him, many of which had migrated from the South up to New Jersey, and in fact, several of them would live with us. And it was always this really interesting life experience of people from different cultures. Maybe it was the customers who aren't always the nicest people in the world, but my father had this ability to, maybe you didn't like the way your pants were pressed, but guess what? We'll do it again. Don't worry. He was just this amazing personality for how to deal with different peoples in different lives. So our family wasn't particularly different than your average sort of, at the time, middle-class family. But there was a lot of love, and there was a lot of strength in our family unit.
Thank you so much. Thank you very much. Before I ask my last question, is there anything else you would like to share?
No, I think you've touched on a lot of really important things. I always like when people highlight the things that matter to me, which are my science, my trainees. I think that, I'm very proud of the fact that I won a couple of mentorship awards over the years, and in the things that I've been trying to do that I hope will help people, whether it's the students, whether it's the patients, whether it's colleagues. So I think you've touched on the things that matter to me.
Thank you. And the last question, which I usually ask to my guests, who I should interview next?
You might try to interview Sean. I mean, he's not easy to nail down, but if you could get him, he's got a great perspective on a lot of things for sure. Who else might I suggest that you interview?
I would like to ask your introduction. I will take a chance now that I interview him.
Yeah, I will. I'm happy to do that. Just
Thank you.
blurb to send to him.
Yeah.
Yeah, and then the only other people, I think the people who have been the heads of the sites of the Parker Institute, my role was, pretty secondary to those folks. So if you can interview a Carl June, or if you can interview a Tony Rebus or Louis Lanier, Crystal Mackel, any of those folks, I think you would enjoy very much talking to any of them.
I will tell them that you would suggest it, so I hope they will not reject.
Okay, well, good luck.
Thank you so much.
Right.
Thank you very much. It was really a great pleasure.
Well, I enjoyed it. Thank you.
Don't forget to like, comment, share, and subscribe to OncoDaily on YouTube. Hit the bell icon to stay updated.